RNA sequencing, or Whole Transcriptome Shotgun Sequencing, is a next-generation sequencing method. Provides snapshot of coding and non-coding transcripts to understand connection between genome and its gene expression, and identify novel features of gene fusion and isoform, as mentioned in next slide. Direct RNA sequencing, performed by sequence-isolated RNA directly, without converting to cDNA. While indirect RNA sequencing or complementary RNA sequencing, performed by sequence cDNA after converting from isolated RNA. Whole transcription RNA sequencing, or total RNA sequencing, sequence all types of RNAs to target both coding and non-coding transcripts, to provide all information about gene expression, as mentioned in next slide. While mRNA sequencing, sequence poly A library, directly or indirectly, to identify expressed coding genes, as mentioned in next slide. Whereas target RNA sequencing, sequence-specific transcript, to study particular genes or pathways, as mentioned in next slide. Otherwise, small RNA sequencing, sequence microRNA, and short interfering RNA, and Piwi interacting RNA, to study gene regulation, as mentioned in next slide. On the other hand, single-cell RNA sequencing, sequence RNA from individual cells, to characterize cellular heterogeneity within complex cell, and identify unknown and rare cell types, and explore changes in gene expression among cell type, and trace trajectory of distinct cell lineage, as mentioned in next slide. RNA sequencing, initiate by RNA extraction and RNA selection to select target RNA, and synthesize cDNA from isolated RNA, and prepare cDNA library to generate millions reads of each sample in process of fragmentation to optimize sequencing of target cDNA, and adaptor ligation to bind transcripts as priming site for sequencing. And index and amplification by add specific sequence or barcode to transcripts during PCR amplification of cDNA to increase concentration of developed cDNA library for sequencing by next-generation sequencing, and analyze obtained data by bioinformatics tools by comparing obtained sequence with reference sequence, as mentioned in next slides. So, RNA sequencing detect differences in gene expression levels without prior knowledge of genome sequence, to detect novel transcripts, and allele-specific expression, and alternative gene splicing transcripts, and post-transcriptional modification, and gene fusions and isoforms and other structural variants. On the other hand, RNA sequencing, consider labor and costly and time-consuming method, because analyzed RNA sequencing data require significant computational resources and experts. Therefore, RNA sequencing, use in mutation, and gene expression, and disease diagnosis, and biomarker, and epigenetic, and functional genomic, and pharmacogenomics studies. Finally, these slides show systems of RNA sequencing, which are iSequence 100 system, and Next Sequence 1000 and Next Sequence 2000. And Nova Sequence 6000 system, and Nova Sequence X series, and another Nova Sequence 6000 system, as mentioned inside slide.